Awesome, we're live. So we're group 4. We're going to do our group problems for lesson 12. I'm Keegan.
Tianna.
I'm Penny.
And we're going to start off. I'm just going to pull my paper up as a guide, and then Tianna will start with problem 1 for us. Present.
Okay, so for question 1, it gives us all these terms, and for A, it says, which of the terms describes or is an example of a regulatory protein? So I found all of these answers from the reading. So what we found is for a regulatory protein was that an activator or repressor in the CAP. For B, it just says, which of the terms describes or is an example of a small effector molecule? We found inducers, inhibitors, allolactase, and CAMP. For C, which of the terms describes or is an example of a sequence found within the DNA? Found the +1 site, the -10 sequence, the LAC-A, and the LAC-Z. For D, it says, which of the following is not involved in the lac operon? And we put none because we believe that all of them are involved in the lac operon. And for E, which of the above is an example of a structural gene? And we found that LAC-A and LAC-Z were both structural genes. And for number 2, it's the vocab, fill in the blank. For A, the lac operon is regulated by 2 inducers, allolactase and CAMP. For B, the binding site for sigma factor in RNA polymerase is called lac promoter or LAC-P, while the CAP site is the binding site for the activator that regulates the lac operon. For C, the enzyme adenyl cyclase converts ATP into CAMP. And for D, -35, -10, and the +1 sequences are found within the LAC-P portion of the lac operon.
And so for E, we found that the LAC-I gene encodes the lac repressor, while the LAC-Y gene encodes the lactose permease. And attenuation can regulate gene expression in bacteria by causing transcription to terminate prematurely. For G, an E. coli strain that contains 2 copies of the lac operon is called a merozygote strain.
Okay, so I'm doing number 3. So you're examining E. coli lac operon. Essentially, like it said, I'll just leave this up there so that paragraph can be read, and then I'll move on down. The question here is determine if the culture conditions described in parts A3 will result in blue E. coli cells or cells that were metabolized by the beta-galactosidase. So for A, I put that there will be no blue because the IPTG is absent, and that is needed for the metabolism. For B, I put yes because you have the IPTG and you also have the XGAL, and glucose is absent, so it doesn't inhibit the metabolism. For C, I put no because XGAL is absent and you need that. For D, I put they're all present, but it's still no because the glucose will inhibit it. And E, no because XGAL is absent again. So only one of them would you actually see the blue E. coli cells after they were metabolized, or would actually be metabolized. 4 is also a little confusing. You have the different genotypes of different E. coli strains, and you have to say whether they're inducible, constitutive, I'll call it, or non-inducible. And so for A, we put that they're inducible. Since they were all the wild type, it was the perfect scenario. For B, non-inducible, the IS makes it so it can bind to the operator, but it can't bind to allolactose. For C, we put constitutive. They both, the chromosome and the plasmid, have the bad operator sequence. For D, non-inducible, again, the IS on the plasmid makes it so the plasmid can't bind allolactose. And for E, you have a couple things. We put constitutive because it can't bind to the allolactose or an operator in the plasmid. And we'll move on to 5.
So this one's the same scenario, but it's asking about, like in the Jacob Bonnet and Party experiment, what colors we'll get. And so you have, y'all can hear me, right? Okay, make sure I'm not muted. Okay. So we have the same kind of factors involved. So it wants to know if they'll, how blue it will be or how blue it won't be type of situation. Is it, the color's blue, right? I can't remember on this one.
Yellow.
Okay, so for the first one, you're going to have 0 color because it is functioning normally because it's all wild type. So for the second one, you're going to have 1, the number 1, for both with lactose and without lactose because the OC we're here referring to that defective operator sequence. So the repressor cannot bind, so it'll I'm having troubles here, sorry guys. So it'll react with that and produce the lactose, the color enzyme there. So for C, you're going to have 2 with the lactose and 1 without the lactose. And again, it goes back to this OC. When you have the lactose, it's going to keep producing twice as much as it normally would. And without the lactose, it doesn't have that extra lactose to start that, so it won't have that extra color. And for D, you're going to have 1 with lactose and 1 without lactose, because here in the chromosome, we have the defective gene. So we have that defective LACI gene, and then we also have the defective plasmid gene, or excuse me, not the P. In the plasmid, yeah, we have this defective gene here as well. So then you only have 1 and 1. And then the last one, you'll have 0 and 0, meaning it won't have any color. The chromosome's normal, but in the plasmid, you have that IS gene that's defective. So that'll cause a little bit of color. Clear as mud, right? That one was tough.
That one was tough.
You guys have anything you want to add to that, or?
No?
No, I don't think so.
Okay.
Just if anyone has any questions, they can just comment
Or
below,
advice.
and we will try and answer anything. So
All right.
we'll see you guys later.
Thanks.